So, peace be upon you all, and welcome to another episode of Seeing God in Biology with Dr. Fuz Rana. First, how are you doing today, Fuz?
Ahmed, I'm doing wonderful. How about you?
All is well, alhamdulillah. And today the topic is DNA computers. So the first part of it is DNA, which is more like biochemistry on more on his side, and the second part is computers. Myself being a electronics and communications engineer with a lifetime in computer systems, so maybe more towards my side. So this should be a fun discussion.
Yes, it sure should. I'm looking
Okay.
forward to it.
Yeah, likewise. So shall we go to the presentation?
Sure. Let's do that. And
Yeah.
what I'd like to do is maybe set the stage
Mm-hmm.
uh, before we jump into the whole idea of DNA computers, just
Mm-hmm.
so people really appreciate the profound philosophical and theological implications of the idea that the molecular machinery that manipulates DNA inside the cell literally is functioning like a computer system. And
Yeah.
if there's been a theme that is kind of reverberated throughout, um, throughout the programs that we've done, it's this idea that information is a signature for the work of a mind, the
Yeah.
signature for intelligent agency. And of course, I think everybody recognizes that our experience is that whenever we encounter information of any sort, we immediately conclude that there's a mind involved. And one of the best examples that or best illustrations of that point I've heard, I think, comes from a philosopher by the name of Richard Swinburne, if I'm not mistaken.
Mm-hmm.
And his analogy is that suppose you're driving, into a community and there's a hillside with a pile of rocks on it. You wouldn't
Okay.
think anything of it. But
Mm-hmm.
if the next day you drove in and those pile of rocks were organized to spell out welcome, you would recognize, right,
Yeah. Somebody's
that
drove it. Yeah, has
somebody,
written it.
right,
Yeah.
somebody is communicating in a message, right, to whoever happens to drive by.
Yeah.
And
Yeah.
and so the point is that when that information really represents a very specific type of order, right, that is distinct from the type of order that you would see, in salt crystals, for example,
Mm.
right? In salt crystals, and that type of order can readily be produced through mechanism, through the regularities of the laws of nature. But information really seems to be in a separate category. And again, we have no experience that tells us information just arises through chance or through the necessary operations of the laws of nature. It seems to be something that is, again, created by a mind.
Yeah.
And
Especially when it's in the right place and the right time, I would say.
right,
Yeah. So a message that looks like welcome at the entrance of a city, okay, does not lend itself to be a product of chance, but
right,
maybe welcome in the bottom of the sea in the middle of nowhere. Well, somebody might have his ideas, but
yeah,
yeah. So I also think that the fitting of the information lending itself to performing a function in its context also suggests something, which I think is something that we see over and over again in systems. Information is not just like
mhm.
isolated from its surroundings or from its context, I guess.
Yeah, that's an excellent point.
Yeah.
And of course, we've talked significantly about proteins. We've talked significantly about DNA, and these are at their essence information harboring molecules.
Hmm.
DNA's primary function is to store information, and we talked about how DNA seems to be optimally and elegantly designed to serve its role as an information storage molecule,
Uh-huh.
and that the information stored in DNA is the information, the instructions, if you will, to build all of the cell's components and really to regulate the activity of those components, both directly and indirectly. And then we also talked about proteins that are also information harboring molecules, where that information that's found in DNA becomes functionally expressed through
Yeah.
proteins. Each of them employs a different kind of molecular alphabet,
Hmm.
And we talked about in the last episode about the genetic code really, being that set of rules that decodes the information in DNA into a protein context. So that translation of information from one format to the other requires a code, requires a set of rules. But again, both of these are information harboring molecules, and the information is digital in the form of discrete units. In DNA, there are 4 discrete units, the nucleotides, and in proteins there are, at least with regard to what's being specified by the genetic code, 20 discrete coding units there. So this is digital information that we're looking at. So on this basis alone, I think a reasonable person could walk away and say, it looks like biochemistry is the product of a mind, because this is information in every sense of the word that we would think of information. And we talked, in an earlier episode about, DNA being used as a digital data storage medium because of its incredible capacity to store massive amounts of information in a very small volume. The information density in DNA probably outstrips almost anything we could imagine in nature. And so, again, this suggests the work of a mind. But something that is also interesting, and this is really setting the stage for us to start thinking about the idea of DNA computers, is that when you have information theorists who've worked on problems in molecular biology, bringing kind of an informatics mindset to biological systems, they've all noted that it's not just simply the mere presence of information that's provocative. It's the fact that there's an organization and a mathematical structure to that information that appears to be identical to human language. And one of the first people to recognize this is I believe a German, if I'm not mistaken, I believe it's a German, or maybe it's a Swedish chemist by the name of Bernd Olof Kuipers, maybe a Swedish chemist. And he wrote a classic work called Information and the Origin of Life. This is what book was probably published in the 1990s, if I'm going from memory here. It sits on my bookshelf. Years and years and years ago, I went through this book. He's writing from an evolutionary perspective, but he provocatively points out that there's a hierarchical organization to biochemical information that is similar to the hierarchical organization that we see in human language. And so this is a table that I've adapted from his book, basically showing that hierarchical relationship that you see. So it's not just simply the fact that there's information that's present in biochemistry. It's that information has an organization to it that is, in some respects eerie in terms of its similarity to the way that we would conceive of and structure information. Now, a few years ago, a team of biochemists were looking at protein structure and ended up doing an n-gram analysis of protein structure. And the net result is that they showed that there is a mathematical structure to biochemical information that's very similar to the mathematical structure of human languages. And the way they demonstrated this was through n-gram analysis. And I'm sure your viewers have firsthand experience with n-gram analysis, whether they realize it or not. And so I'm going to make my feeble attempt as a biochemist to kind of explain n-gram analysis. Then you can come in and rescue the mission.
Sure.
But and please feel free, of course, to jump in. But the idea behind n-gram analysis is that whenever you're writing a text, for example, it's remarkable how the software will actually suggest the next word that you should probably want to type. It doesn't always get it right, but if you actually have apps that are more sophisticated, they will learn your behavior and begin to suggest words based on what you typically type. But what's happening is that there is a set of grammatical rules that indicate the probability of what the next word is likely to be if you're typing a word.
Yeah,
And
yeah.
so,
We all see it on predictive text when we're messaging somebody or yeah,
right.
yeah,
Yes. Right. And so so that approach, or that the determination of that probability based on grammatical uh, rules and usage is called an n-gram, algorithm, I guess.
Hmm.
And so, here if it's showing you that if you're going to type the cat in the hat, if you type the cat, it's it's likely that you probably are going to type in the hat, and so it's going to suggest to you in the hat as
As you go.
you're right.
Yeah,
And so and when it comes to n-gram analysis, there are different ways in which the probability can be determined: a unigram, a bigram, or a trigram, which just simply means you're just simply using a single word to determine the probability of the next word. Bigram is that you're using 2 words to determine the probability, and trigram, you're using 3 words. And of course, a trigram is probably going to give you a greater level of success than a bigram or a unigram.
yeah,
And so this slide here is illustrating the difference between kind of a unigram, a bigram, and a trigram. I'm not sure if it's easy
yeah.
to see that in the diagram, but anyway, that illustrates the concepts. Now,
It essentially says that the accuracy of the prediction of the next word gets better if you consider more words to try to predict it. So one word can lead to one word, but 2 words can lead to the next word in a better accuracy, and 3 and 4 as we go. Yeah.
right. So now what's interesting is there was a research team, again, a few years ago, that performed an n-gram analysis on protein structure. Now, when we think about protein structure, of course, ultimately, the information that dictates protein structure is encoded in DNA,
Yeah.
right? And so what they argued here is that instead of treating the entire protein as corresponding to a word
Uh-huh.
like, let's say, in the English language or in Arabic, what we can do is treat protein domains as words. And what I mean by a protein domain, this is referring to the 3-dimensional structure of a protein. And biochemists have noted that many times proteins' 3-dimensional structures consist of these independently folded units that oftentimes are functionally independent as well, that
Yeah.
actually exist as discrete units in the protein structure. So here's a structure of a protein that has 3 domains that are depicted by different colors: blue, white, and green. But when you look at the way that they fold, these look like they are actually discrete units, right?
Yeah.
In that are contributing to the protein 3-dimensional structure.
But
And
this is essentially a single peptide chain.
yes,
It's coming from the transcription and translation of just one gene or one DNA strand, let's say.
yes,
Yeah,
exactly.
on one DNA sequence between
Yeah.
2 terminators. Yeah, all right.
And biochemists have discovered that there are domains that recur in biochemistry. They're not, so it's not like each protein has unique sets of domains, but there are domains that again recur.
Uh-huh.
And so this slide shows 2 different proteins, each of which have 2 domains, but these proteins share the same domain. So the same domain is occurring in 2 different proteins. And again, this is because domains oftentimes have discrete functional properties.
So
So
the reddish or pinkish one, it's essentially the same one, but it's used once with this protein and once with the other one. Yeah.
yes, exactly.
Mm.
And so so the idea here is that you can combine domains in such a way to create all kinds of different types of proteins. that would have different structural and functional properties that would be kind of a mix and match combination of a library or a catalog of domains. And so the researchers asked the question, are there a set of rules that actually correspond to how these domains are combined, or are they just randomly combined, and through evolutionary processes? And what they discovered is that
it's like they're looking if there is, like, for example, a kind of grammar in this protein sentence or something.
Exactly. That's exactly the word that they use.
Right? Really?
Right.
Wow.
And what they discovered is that only 5% of the possible domain combinations actually occur. So 95% don't ever occur in the proteins in nature. And from that, they conclude that there must be some kind of grammar, some kind of rules that are determining how these different domains combine.
Amazing.
Right? And so then they went off and they did it.
So, for example, in your example, if we're starting by then the next thing has to be a noun, for example. You cannot say
yes,
the and then a verb. If it's a the, then it has to be a noun or something. Yeah.
Yes, exactly right. And of course, part of the challenge then is to decipher what that set of rules are, because that then leads to fundamental understanding potentially of protein tertiary structure, which would allow biochemists to design, in the lab, kind of novel proteins.
If you need to speak a sentence that makes sense in a certain language, it's not just about knowing the word, it's also knowing how this language constructs sentences, which means that you need to understand its grammar.
Exactly.
Yeah.
And so anyway, this was uncovered through n-gram analysis, and then doing kind of a mathematical characterization of the biochemical language relative to human languages. They discovered something rather remarkable, that the similarities between natural languages and genomes are apparent when domains are treated as functional analogs of words in natural languages. And so what they've seen here is going beyond what Kuipers discovered, which is this hierarchical organization. There's a mathematical structure to biochemical information that, again, is eerie in terms of its similarity to human languages as well.
So he's actually using the same word here in the title: grammar of protein domain architectures. Wow.
Yeah. So fascinating insight. But this is really, again, setting the stage for how we want to think about, I think, DNA computers. Because what biochemists have also discovered is that the fundamental operations of computer systems are identical to the way the cell's machinery manipulates DNA, whether it's in replication, transcription, or in DNA repair. And to appreciate this, and I may need to rely on you to help me, work through this, but because again, this is a biochemist talking about computer science, so be forewarned. But to really appreciate why this is the case, we need to consider the work of Alan Turing, who many people consider to be the father of modern day computer science. And there's some people that argue he built the very first modern day computer during World War II, as part of his work with the British government, looking to crack the German Enigma code. But anyway, that work was built on his theoretical work, I believe, in the 1930s, where he invented these abstract machines to help think through how you could perform complex computations with machines. And so he develops these things called Turing machines that consist of an input and output and a finite control. And so this is a cartoon that illustrates the Turing machine, where you've got a digital input that's being fed into this hypothetical finite control that operates on that digital data according to a very restricted set of operations producing a predictable output.
Yeah.
And Turing's genius was to realize the output of one machine could be the input of another. And by combining different types of finite controls together,
yeah,
you can perform complex computations. And so that's, in a sense, what a Turing machine is. And,
and
is there anything that you need to add to that?
Yeah. So the funny thing is, he actually his Turing machine actually used to alter the input. So the tape that's coming in from the right is altered, and you get at the end of the other end another tape where the very same material now has a different set of information that has been processed by the machine. And I think the abstract thing is that Turing machine just keeps taking in input. It just goes on and on, and the concept itself doesn't lend itself to do anything really beneficial in life. But it turns out that the most strange ideas, after we get used to them, become crazily useful. But then once I'm looking at this, it actually is so striking that it resembles a DNA strand or something.
Yeah.
Since we are in this context, so
Yeah.
the matter of and by the way, people think of computer memory as if it is this vast, maybe three-dimensional virtual space that sits inside the computer. The reality actually is that computer memory is essentially a string of bits, and it's to access a certain byte or whatever word of information in a computer memory. It is a sequential set of addresses. So you start from address 0 until address no matter how big. Essentially, there is a counter, which is the address of the byte. So the computer memory is like a very long street, and with very a continuous set of one-story houses there, and each house can have a person or a no person, and then each set of houses would give you a certain representation of a piece of information. So actually, this machine, as simple as it looks, with a machine in the middle doing a very simple operation, and then you can add them in sequence or make them cooperate together, is essentially at the fundamental level what a computer, a modern computer is. It hasn't changed actually that much. Actually, the conceptualization that Turing had in mind is not even possible to realize with our current technology, because it has this infinite aspect to it. So something cannot become an ideal Turing machine easily, given that we have finite materials in this universe. But the amazing thing is that when you look at this and then you look at the DNA, which is a strand, a linear thing, and then out of this linear thing, you can generate another linear thing through some operations, which is the RNA and then the proteins. And then they fold onto those three-dimensional structures. Just like we're looking at this screen and we are seeing a two-dimensional plane on which three-dimensional structures are represented that represent ideas. And all of this stuff is coming from this linear program that's running in a linear memory on something that is conceptually like this. Actually, to me, it is a striking resemblance between what is the best product of our minds for doing computation and what is actually implemented inside our own bodies. This is just what comes to mind.
Well, and it's your anticipation is uncanny, because in a sense, when you look at a process like DNA replication, it's in a sense, again, in a very real sense, a computer operation, where the DNA again contains digital information, and that digital information is operated on by proteins, which you can think of as the finite controls that enzymes have very limited capacities, and so these individual proteins are operating on DNA in various ways, where the DNA is the input and then becomes the output as well.
Yeah.
And you, by combining, the different Turing machines and their operations, and in a sense, the operations are occurring sequentially in time, you're able to perform a very complex process through a combination of again, enzymes working together as, in effect, finite controls. And so this is exactly the way in which, a Turing machine is operating, that you really are looking at a combination of biochemical Turing machines that have been combined in distinct ways to create, in effect, a computer operation inside the cell.
Actually, we can see a direct analogy between what DNA replication is, and when you have a file, for example, on your hard disk, and you're creating another copy of the file in another directory, or you're sending the file through over the internet to somebody else, or you are uploading it to YouTube, for example, or whatever. In all of those processes, there will be those little conceptual Turing machines that are copying the information, maybe from the hard disk to another place in the hard disk, to another place or across the globe, whatever, and essentially actually doing very similar things to what this process does. So it's copying, it's verifying the copy is correct, it is reassembling the very bits into bytes into a complete file at the end, it is terminating the file, it's giving it a place in the directory so that you can find it, etc. So it's really amazing how really this very sophisticated process of DNA replication is what we can see. Of course, somebody will just type copy file XYZ to a directory AKZ, but essentially at the fundamental level, it's the resemblance is really striking.
Yeah, and we have a video that will show that really, is DNA replication in action. And as the viewers watch the video, just think about each of these individual proteins that are part of the replosome as being, in a sense, Turing machines that are working collaboratively. But when we look at DNA replication, we really are looking at a computer operation at its very essence.
Absolutely.
which is pretty mind-blowing when you think about it.
Yeah, let's have a look at it. Yeah. Okay, I guess that was quite an amazing video, and the amazing thing about it is that this specific machine is actually not only a digital machine, but it's actually even a mechanical machine.
Yes.
So imagine the computer inside had to do all those mechanical work to create this thing. Because the amazing thing is that this is not just electrical signals on a tape or optical drive or magnetic drive or USB. It is actually chemistry. It's actually chemicals put together and stitched together and terminated and all the things that are maybe easy on the electrical or magnetic level or optical material are very sophisticated in terms of biochemistry, because biochemistry is very difficult to work with, I guess. If I ask you as a biochemist, is it like the easiest thing in the world to create a biochemical reaction or not? Maybe you can comment on that, so people would appreciate how difficult it is of an environment that those machines are operating in.
Oh, it's extremely challenging, to produce to conceive of from scratch a biochemical operation is unbelievably challenging.
Yeah. And I guess we discussed before, like a biochemical reaction, if you just leave it on its own to happen versus getting the right enzyme in the right place and the right time to make the reaction happen. And I guess the order of magnitude of the time needed was like tens of millions of times or something. It takes like a fraction of a second under the effect of an enzyme, and maybe, I don't know, 10 million years if the enzyme was not there. It was totally amazing.
Yeah, I mean, most of the chemical reactions in the cell are going to be relatively speaking, very slow reactions on their own.
Yeah.
And enzymes will accelerate those reactions, in some instances, probably close to 1 trillion-fold. It's, it's an incredible acceleration that's taking place. So you don't really have the capacity for life without enzymes. And this is one of the problems with appealing to kind of a metabolism first approach to the origin of life is you might be able to produce these autocatalytic reaction cycles, but they're going to be very slow.
Yeah.
And not fast enough to be of any appreciable value for an emerging protocell, under evolutionary conditions.
And I guess getting back to the example that you gave in the beginning about the stones writing welcome at the entrance of the city or something. And then now here's this cell that wants to replicate. It's doing everything that's in the context of replication. And then guess what? The welcome word comes in the right place, in the right time, to the right, in the nucleus, during the replication process, and comes there and is serving the purpose of replication. So nobody can say that these enzymes are there by chance or they evolved from, completely haphazard processes. I think it's totally absurd.
Yeah.
Yeah.
And so just as a quick summary,
Mm.
you can think of, again, DNA, which is digital information, as both the input and the output, and then the enzymes that are manipulating DNA is the finite control. And of course, you even have error correction systems built in. So this is which in and of themselves are Turing machines. So you've got this
Nesting.
very complex system that ensures not only replication, but high fidelity replication is taking place. Now, one of the things that I find interesting is that when an argument like this is presented, somebody who's a skeptic, oftentimes they'll say, well, these aren't really computers. This is just a metaphor, that biochemists are using, for illustrative purposes, just to help us grasp the concept. But you're really going overboard to say that this is actually a computer system. And in response to that, I like to point out that the operation of DNA replication is so much like a computer that it inspired a computer scientist by the name of Leonard Adleman, who's shown here in this picture, who's at the University of Southern California, to actually invent DNA computing. So he's considered the father of DNA computing.
Wow.
And so the idea is that these are wet computers that are built from DNA and the enzymes that manipulate DNA, and they're housed in little tiny Eppendorf test tubes that are this big. And these computers are more powerful than the most powerful silicon-based computer system that we've built. They are more powerful than supercomputer systems. Wow. And this is becoming a fairly well-developed field now in nanotechnology, DNA computing. And in fact, Adleman, to illustrate the power of DNA computing, solved something known as the directed Hamilton path problem. And this is also known as the traveling salesman problem. And so the idea is...
It's an optimization problem.
Yes. So the idea is that you have n number of cities, and the salesman has to travel to each city, but he can only go to the cities one time. And so the question is, what is the shortest route he would take to go from city to city to city to city? And when it's a few cities, it's fairly easy to solve. But once you start adding the number of cities, it becomes an exponentially difficult problem, where I think a standard silicon-based computer is really not capable of solving this directed Hamilton path problem, as I understand it. And so
it takes more and more time. It grows so much. I think it can be subject to solving maybe with an advanced quantum computer, if quantum computers work at all to really solve those kinds of problems.
Because the problem is that once you determine what you think to be the shortest path, the only way to check that is you have to go through every possible permutation.
Yeah.
to evaluate, is this actually the shortest path? And so what Adelman did is that he had blocks of DNA of differing sizes, where the size of the DNA block corresponded to the distance between two cities, right? Does that make sense?
Uh-huh. Uh-huh.
Right. So if you have, 10 cities, you can, whatever that, you can figure out the, computationally, what is the number of DNA blocks that you would have, where you're looking at what is the distance from A to B, that would be one DNA block. What's the distance from B to C? That's another block. And the size of the blocks then are going to correspond to those distances.
Okay.
And then
the size is then the number of nucleotides in each.
Yes, exactly. So if it's 20 miles, then it might be 20 nucleotides. If it's 10 miles, it might be 10 nucleotides, as an example.
Okay.
Right. And so then you just simply take these DNA blocks, you throw them into a test tube. You have some kind of enzyme like a ligase that will just randomly join them together. And then after a period of time, you can essentially run a gel and identify which is the shortest piece of DNA. And that turns out to be
the
path, the optimal path.
Okay.
Right. And so, because what happens is you can create vast numbers of molecules that you would, put into the test tube, and they're all going to react simultaneously.
Yeah.
So in effect, you have this massive parallelism, this massive capacity to store huge amounts of data within DNA, and you're operating with a very high level of energy efficiency.
Wow.
And so in virtually an instant, the problem is solved, right?
How would he find the shortest one?
You would have to, once you've added all the DNA pieces together in the test tube, you let the ligase do its job.
Yeah.
You would have to run a gel, what's called a gel. So where
it will sort them. Yeah.
Yes. So you'd sort them based on size. So the disadvantage is
it's like a kind of precipitation where if it's heavier or something, it goes up or down. So
right.
Yeah.
Usually with the gel, it's kind of like a meshwork. And so things that are very large will go very slow. Things that are small will go more quickly through the meshwork.
Yeah. Okay.
Right. And the concentration of the gel will determine, the range of sizes that you can separate.
Yeah.
And so anyway, so it's very, the drawback is to run the gel, and then to isolate that band and then to sequence it, takes a few days. So while, it's a very rapid,
reaction
system, the readout is very cumbersome at this point in time. But the point is that, for our purposes, is that this is an incredible achievement, right?
Yeah, because it's unsolvable anyway on the other side.
Right. But and again, people are looking at how do you actually, develop much more sophisticated capacities to solve, real life problems. What are the types of, computer algorithms that you can develop, and if you can develop modules, you can then combine modules together to do very interesting things. But if you have something like the Hamilton path problem that is essentially you're unable to solve with conventional computing, maybe it does take you a week, to do the work to read out the information, but you've solved the problem, right?
Yeah. And there is a solution at the end.
Right. And really the limitation of DNA computing isn't the actual nature of the computer itself. It's essentially our limitations, technologically speaking, as human designers, right?
Yeah.
Right. So,
if we have something that's faster than the gel, or that can be easier to pick up the solution, then we would have known faster. Yeah.
Right. Exactly.
Yeah.
Exactly. But from a theological perspective, to me, what's significant is that these DNA computers in the cell are not metaphors for a computer.
Yeah.
they actually are computers, because you have scientists that are building DNA computers using the same machinery as what's found inside the cell. And so the right way to think about DNA replication or transcription or DNA repair is really that these are computer operations. And in fact, we could even include translation at the ribosome as ribosomes as being another type of computer operation, which is even far more sophisticated than what's happening with DNA replication, because you're actually now operating in two different formats.
Yeah.
Right.
Yeah.
So,
Absolutely.
so to me, it's absolutely mind blowing. And this is in an interview, Leonard Adelman said this: the most important thing about DNA computing is that it shows that DNA molecules can do what we normally think only computers can do. This implies that computer science and biology are closely related, that every living thing can be thought to be computing something, and that sometimes we can understand living things better by looking at them as computers.
Okay.
So it's, just a incredibly provocative statement, on the
Absolutely.
part
Yeah.
of Leonard Adelman.
Yeah.
Now, to me, this, much of what we've talked about over all the episodes that we've recorded, really, I think, can be condensed into this watchmaker argument of William Paley, right?
Yeah.
Where Paley is making a comparison between a watch and a rock. In his day, you could argue that a watch was probably the pinnacle of engineering achievement. And Paley's point is that, look, when you pick up a rock, we all understand that this is formed through natural processes.
Yeah.
But when we pick up a watch, we realize that this is, of course, the work of a watchmaker. And what is it that leads us to that conclusion? And it's the fact that the watch has these attributes that reflect essentially what we would expect to see if a designer was creating the watch, right? And so when Paley surveyed biological systems and what was known of them at that time, he concluded that these systems have much more in common with a watch than a rock. And so if we see these same kind of designs in humanly produced systems, we would conclude a designer. Why should we not conclude a designer in biology? And, what we're talking about over these past episodes is really an opportunity to kind of revitalize the watchmaker argument in a biochemical context. And so we know that where computers come from, right?
Yeah. Absolutely.
And computers are the pinnacle of our engineering achievement. And so when we see that there are literally computer systems that are at the core of biochemical systems, and that these computer systems are eerie in their similarity to how we would independently design, computational machines, it's absolutely eerie. But then on top of that, we've talked about the fact that DNA is this storage medium, right?
Yeah.
That we've talked about, the idea that embedded in DNA is this even bit parity code that's an error detection system, identical to how we would do it. There's there's information that has the same structure as human language. There's a code, right, that displays the properties of how we would construct codes, right? And it's highly optimized. It's when you put it all together as a package, you've got a very powerful, I think, weight of evidence type of watchmaker argument, where it's not just a single feature of the cell, but it's an ensemble of features. And of course, on top of that, we could argue that there are machines that are involved in these processes that bear eerie resemblances to the machines that we would build. As you're saying, that this isn't just a string of electrical pulses or the absence of electrical pulses, right?
Yeah.
This is actually a three-dimensional system where there's the Turing machines literally are actually three-dimensional machines that in many instances are identical to the machines that we would build. And so, it's it's instead of its turtles all the way down, it's design all the way down. Right?
Yeah. Actually, I have an insight. Maybe I would like to take your opinion also about that, about the watchmaker argument. Um, so if we think what makes a watch, it's not its sophistication, but maybe it is that this sophistication leads to this object telling time. So if you find something that looks very sophisticated but it says nothing to you, it doesn't appeal to your conscious as saying anything. It's just something that is sophisticated, something that has been carved by the wind or something. It looks very sophisticated for any reason, but it doesn't say anything. But the amazing thing about the watch that makes it compelling to call for a watchmaker is that the watch actually tells time.
Right.
And we know that it tells time because when we look at it, we have a consciousness that relates to the concept of time, and we see that this object, if we have not seen the watchmaker and have not seen him make it, and we find a watch and we look at it and it's working and it's telling time, we know that it's telling time. And then it strikes me that living things, even if we think of them as computers or even if we think of them as machines, because why do we think computers are doing anything? Is that because they have programs? And if you get a computer that's a piece of metal and you power it up and you just keep what is in its memory, a complete random glitches, a series of random glitches of ones and zeros, it will do nothing. Nobody would appreciate if human civilization is wiped out and there is this one remaining laptop that's discovered 2,000 years from now, but it has no programs on it, and somebody figures out that it's working with electricity and powers it up, and it shows complete rubbish on the screen. He would not appreciate that this is a computer because it will not tell him anything. So what makes a computer is a program that tells something. And I think what makes the issue of life compelling, that it must have a creator, is what also makes a computer compelling, that must have a manufacturer and a programmer, is that life says something. Mm-hmm. If there's a bird, the bird is saying something in its environment. Its life has meaning. It grows up. It has little eggs. They hatch. It hunts. It has a community. It has language. Each new individual says something in its context that its context appreciates. Mm-hmm. So the bug comes, it says something to the plant, it pollinates the plant, it completes the circle of life, it dies. The bacteria say something. They decompose the insect becomes part of the soil, and it says something to the plant once more because it eats it up, and then a human being comes, or a monkey comes, or a bird comes and eats the plant. The whole thing is in complete harmony. It is really frustrating that anybody would look at this magnificent system that not only is a magnificent system, but each piece of it makes sense in the context of everything else. But it also makes great sense in the context of a conscious being like us, that when we look at a flower, we say, wow, it speaks beauty. When we look at a fish, wow, this thing knows how to swim. This thing knows how to navigate. This thing knows how to migrate. What a beautiful sound of a bird that is. It sings. And then we look at each other as humans, and we have language, and we have ideas and innovations, and we have religion, and we have rituals, and we have social constructs. And then somebody comes along and says, well, guys, guess what? This is like, it's like a pile of rocks that has no order. It's not saying welcome. It's not saying anything. And it really strikes me that anybody who would really appreciate what it means to be intelligent would accept that as an argument of any sort. I hope that my idea is
no. I think it's a very powerful point that you're making. That because the William Paley's watchmaker argument is oftentimes very readily dismissed by skeptics. But the more that I think about his argument,
Yeah.
The more I've come to appreciate his genius, right? He tends to be lampooned, but it's really unfair because he really was onto something absolutely profound. And the fact of the matter is, everybody who's making a design argument today is in effect owing a debt to Paley, right?
Yeah.
The idea of irreducible complexity is essentially the same kind of argument that Paley was making, or Bill Dembski's idea of specified complexity.
Yeah.
In my book, The Cell's Design, I talk about an intelligent design pattern, right? Doug Axe, in his book Undeniable, talks about functional coherence. These are all basically saying the same thing that Paley said, 250, 300 years ago, or something like that.
Absolutely. Absolutely. And what's the funny thing is that when I'm not sure if I should even call them skeptics, but this category of people who are in denial, let's say, not skepticism, sometimes it strikes me that their argument is, well, guess what? There is no watchmaker because this is not actually a watch. This is essentially what they're saying.
Yeah.
There is no design in the human body, for example, or living things, because this is that you are calling computers are not actually computers, and this is that you are calling an organism is essentially not actually even anything that knows that it exists. It's all an illusion. Yeah. At its core, their fundamental argument is there is no watchmaker because there is no watch. Just like nobody has written the word welcome because the word welcome does not exist. It's only in your mind.
Yeah.
This is their argument. And the frustrating thing is that we're fighting with them about the watchmaker argument while they do not acknowledge the watch in the first place. This is the frustrating part about it. Let's first agree that there is a watch and then talk about the watchmaker. But the watch is very obvious. It's telling time every second. So how come it's not a watch? This is the question.
But this is where I think Adelman's insight and
absolutely
and creation of DNA computers, it's not like he's co-opting these molecules inside the cell and using them in ways that the cell doesn't use them. He's using them identically, in the ways that the cell uses them, to create these interesting, computational processes in the test tube. Absolutely. But he's really highlighting these are computers.
And even you have given before the example when we talked about DNA storage, about scientists creating actually hard drives where he can actually store a book or a picture or a JPEG on DNA, right? So it's showing them its storage.
Yeah.
So it just happens over and over again.
Well,
what do you think? Does this say that there is no end for denial? If you want to deny, you can just deny anything.
Yeah, I think so. And it's interesting because I've heard people talk about the fact that, but a cell is able to replicate, and of course machines that we build can't replicate. But if you then go to and look at the ideas of what's the guy's name now? This is the struggle growing old. You can't remember things. The von Neumann universal constructor, right? So he conceives of this machine that is able to build any machine, and in fact it can even build itself, and that there is this switch where the machine uses its instructions designed to build other machines to then in turn use those instructions to build itself. This is exactly the cell.
Yeah.
Right. And Paul Davies is, in a journal article a few years ago pointed out that in a sense the cell is a von Neumann universal constructor. And so even though people might say, well, again, we don't build machines that replicate, we don't, but according to von Neumann, that's the ultimate engineering project, is to build self-replicating machines, right? And so it's analogous in a sense to to the Turing machine, which, was this crazy idea that Alan Turing had, that then later, as you said, turned out to be incredibly useful, for building machines that then turn out to already have, that already exist inside the cell.
Absolutely.
So,
yeah. And let me close with this. In one debate that I had, somebody threw at me, well, you can't give examples for cars or computers or whatever because they're not self-replicating. And I just told him, actually, the hope that space colonization will happen someday is through self-replicating machines. That you will send a machine to another planet, it will self-replicate, start consuming material to build other machines, and then build a civilization on that planet, terraform the planet, and then you can arrive. And this is what nanotechnology is trying to achieve, etc. So we are trying, and we actually do have self-replicating machines. If you can consider that you can model, you can build a computer program that actually can replicate itself, okay? And you can allow it while replicating itself to make errors, okay? And then the question is, and you can give it a huge amount of memory and a very fast computer, and you can just leave it replicate there. And then my question is to any honest computer scientist or honest biologist or honest computational biologist out there, show me an experiment where you start with the very basic computer program that you can make it as a resemblance to Luca or something, the lost universal common ancestor, the hypothetical lost universal common ancestor, and show me what it becomes after a trillion iterations. Do whatever number of iterations that you would like that will come close to the number of iterations that you think the first cell went in, and show me what it does. Will you have a chess playing program? What will you have? And it doesn't exist. This example will never yield anything, because you need to put it in a condition where nobody is directing it, where it's just sitting there, processor is just copying it, allowing for some errors. There is vast, huge amount of memory and very fast computer, and show me what it becomes. Will it become a watch that tells time? And from where will the consciousness of understanding that this is a watch and that will understand the concept of time come?
Yeah.
And since there is no answer to this question, no matter how advanced our computer science today is, including AI, including all of other algorithms, nothing works if you're not directing it. And then it says that even if there is a solution to abiogenesis, then the question is, who has directed it? Right. And it all lends itself to, there is God behind all of this. There is no other solution, I guess.
Yeah. Yeah.
Yeah. So, Fuz, this has been wonderful. DNA and computers in the same episode.
Yeah.
And as usual, we all owe you a great thank you for showing and sharing this with us. And to everybody who have watched this, please help us spread the message, share it with everybody that you love, and give us your comments and your questions. And we might as well do an episode where we'll put together questions and answer them. And thanks a lot, Fuz. And see you again next week. What will we be talking about next week?
I think we're going to be talking about biochemical finite machines.
Wow,
which is another type of machine, another type of mechanical computer, if you will, that are found in the cell.
Looking forward to it. And thanks to everybody. And see you again soon. And peace be upon you all.